Here's part one of a discussion of the cell membrane. We actually began this discussion back in the osmosis lab. We talked about the structure of the cell membrane, about the involvement of phospholipids and proteins and short chains of carbohydrates. So if you review your notes from the beginning of the osmosis lab back a few weeks ago, that would be helpful. I will also post in the folder that this video is in some drawings of the so-called fluid mosaic model, something we discussed back in the osmosis lab also, describing the various molecules that are there. I'm not going to try to draw it here. I'll post a picture of a much better quality drawing than what I could possibly do myself. We also, at the time of the osmosis lab, discussed some functions of the cell membrane. And there are many. The one that we're going to focus on in the immediate future here is that the cell membrane is going to help regulate passage of materials in and out of the cell. It's a gatekeeper, so to speak. It's a barrier, a border that separates the inside of the cell from the surroundings, but within limits, the cell membrane can make decisions, so to speak, about what can get in, what can get out, what can't, etc. And in this video and in one or two more following, we're going to take a look at some of the ways that the cell membrane does that, and also what happens when that doesn't work properly. So this is page 43 of the lecture outline book. I'm going to switch to page 44, and that's a drawing. Again, I will post more drawings on Blackboard. But let's get to mechanisms of membrane transport. And again, by this I mean ways that the cell, specifically the cell membrane, will make decisions about what gets into the cell and what gets out of the cell. We're going to look at a few examples of what are called passive transport mechanisms and a few examples of active transport mechanisms. So first, it's important to say what these two terms mean. A passive transport mechanism is a mechanism by which some object, a molecule, an ion, let's say, will move across the cell membrane, either getting into or out of the cell, but that movement does not require that the cell itself spends energy. So I'm going to write something like the cell does not provide the energy. Anytime something moves, energy is involved. Whether it's passive or active, energy is involved. So to say that passive transport means that energy is not involved is simply incorrect, and sometimes that is the way things are written, and that is not true. The catch here is a passive mechanism of membrane transport means that some object is moving across the membrane, but the cell itself is not providing the energy that is needed for that movement. In an active transport mechanism, and we'll see a few of those as well, the material in question, the molecule, the ion, whatever it is going to move across the cell membrane just like it did here, except in this case the cell has to pay, so to speak. It's the cell's responsibility to provide the energy to make this move. So here the cell provides the energy. I use an analogy. It's like saying I could go on to Amazon and order some product, and if I get free shipping, that's passive transport. If I have to pay for the shipping cost, that's active transport. Whether or not I paid for it doesn't mean that it happened for free. In other words, even when I don't pay, somebody had to pay. Someone paid the UPS driver to bring that package to my house. But if I didn't pay that fee, that's passive as far as I'm concerned. If I had to pay that fee for shipping, that's active. And that's analogous to what's happening here when the cell experiences movement of materials across its membrane. So what we should do here is start taking a look at a couple examples of passive transport and a couple examples of active transport. And the picture that you see here is sort of pre-drawn. It's supposed to be part of a cell membrane. Each of these little things here is supposed to be a phospholipid molecule. And again, if you review your notes from previously in the picture that I'm going to post here, you know that phospholipids are typically drawn as the little circle with the little two tails coming off there. These are fatty acids. This is the hydrophilic portion of the molecule. And again, as you might remember, the fluid mosaic model states that the cell membrane is made largely of a phospholipid bilayer or a double layer. So here's phospholipids and another one and one after another. So what I've drawn here and in future panels is part of what is really a whole cell membrane, and we're just catching the corner of it. So this is the inside of the cell and this is the outside of the cell, just so that you're oriented to that. So let's talk about passive transport mechanisms, and the first we're going to discuss is simple diffusion. Simple diffusion can be defined in sort of a textbook way. I'll do that. It's the movement of material. That could be a molecule, it could be an ion, whatever. Movement of material from an area of higher concentration to an area of lower concentration. And so let's talk about that without even mentioning cells. Let's say I have a bottle of cologne, and I take off the cap and I just let it sit on a table. Given enough time, the whole room's going to smell like that cologne because I have this point source, so to speak. Here's the bottle of cologne, and here's the room. And molecules of cologne are going to start to evaporate, and there's a very high concentration of them here. But just by the random movement of these molecules, they're eventually going to wind up getting to various other places in the room. So they're moving from an area of high concentration to areas of lower concentration. This is simple diffusion. It has nothing, in this example, it has nothing to do with a cell. It's just something diffusing around a room. This happens all the time. So notice that you don't have to be describing diffusion through a cell membrane. It can simply be from any area of higher to lower concentration. Now for today's discussion, of course, we're interested in how that relates to cell membranes. So can a material diffuse from a higher to a lower concentration through a cell membrane? Certainly it can. Let's say, for example, there's a particular molecule. I don't even care what it is right now. We'll just say each of those little orange dots is that molecule. And let's say outside the cell, there's a particularly high concentration of that molecule. And inside the cell, there's a much lower concentration. Simple diffusion states that materials move from higher to lower concentrations. So you're going to get a net movement of this molecule from here to there. If there were a higher concentration inside than outside, the movement would be in the opposite direction. Now there's one caveat here that's important to note. This membrane has to be permeable to this material. In other words, this material has to be able to fit through that membrane. That's not always true. If this molecule is, say, exceptionally large or has a chemical nature that might prevent this, even if there is more of it out here, it won't diffuse from here to there. In other words, there are cases where the molecule itself simply can't fit its way through the membrane, and it doesn't matter whether there's more here or not. It's not going to move. This is a barrier. But assuming that this stuff can get through the membrane, assuming that the membrane is permeable to this particular material, it's going to move from the higher to the lower concentration. Now, just to add one more variable into this, what do you think would happen if there was, say, 5 times as much of this material here than there? Well, there would be a certain rate at which this material goes from here to here. If instead there was 100 times as much here than there, it would still go in the same direction, but much more quickly. In other words, the rate of diffusion is dependent, among other things, is dependent upon how much different these two concentrations are. If there's twice as much here than there, it's going to go pretty slowly. If there's 100 times as much here than there, it's going to go a lot more quickly. And that's important to note when we start applying this to real life examples. We're just using a made up molecule here right now, but we need to keep that in mind, that the rate of diffusion across this membrane is dependent on how big the difference is in concentration here to there, one compartment to the other compartment. There's an easier way to say that, and that way involves using a word that we haven't used yet. That word is gradient. I apologize, that ink doesn't really show up well. I won't use that color anymore. Let's try this. There we go. The word gradient, and this is on the key terms box on the previous page. When you talk about a gradient, you're talking about differences in concentration. So I'll go back to this cologne example. If I consider this room and a bottle of cologne that I just opened, there's a gradient in cologne. If I compare the open mouth of the bottle to, say, over here in the room, there's more of this material here than there is there. There's a gradient. The gradient just refers to the difference in concentration. There might be 10 times as much here than there, or 100 times as much here than there, or 1,000 times as much here compared with there. Either way, there's a gradient. So I can talk about the same thing with respect to this cell membrane. If there's 100 times the concentration of this molecule here than there, I've got a pretty steep gradient, and that's a descriptor that you can use. You can talk about how steep the gradient is. You can talk about the magnitude of the gradient. If there were only, say, 2 times as much of this material here than there, that's a much less steep gradient. It's still a gradient, it's just not as steep. If there was the same concentration of this material here than here, there's no gradient. So this is a handy word to have when we start discussing movement of material across membranes and concentrations of material on one side versus the other. The existence of a gradient is something that we're going to be focusing on in future examples. In this particular picture, there's certainly a gradient of the little orange dots. There's more of them out here than there is in there when I'm comparing this space to that space. And when a cell membrane is involved, it's very easy to know. I'm talking about the outside of the cell versus the inside of the cell. So that's a quick summary of what we call simple diffusion. I'm going to stop the video here because I want these videos to be fairly short because they're easier to upload and easier for you guys to watch in little bits. And I'll film another video about some other passive transport mechanisms.